Plant breeding is a comprehensive and complex science, with a lot of disciplines working together to form a final end product, a new variety. However, with a few basic principles, plant breeding can be easy. And that's I want to show you. Breeding is easy. You have just to create the variability and in the second step to find the best candidate. Imagine we have one very good line in the field with poor baking quality, unfortunately, and we have the other one with perfect baking quality. Now the idea is to make a cross of both, many progenies, to have the probability increased that one line combines both traits, and in the second step we try to find it in multi-location, multi-year trials. That's all. But to be efficient, we need to know more. Breeding aims to improve traits like grain yield or baking volume. To do so, we need some important figures to be able to do it. First, we need to know is there any genetic variance available, because breeders work only on genetics. Second one is the heritability. This measures how much of the visible variability in the field comes from genetics. Third is correlation of the trait to other traits, because when the correlation is negative to an important other trait, it gets complicated to breed for both. And finally, we have to measure a lot of candidates very fast in the field. That means we need rapid test method for the traits. Keep these 4 things in mind when you think about to optimize trait breeding. Heritability is the king of formulas we need in plant breeding. It quantifies in the visible variance in the field the amount of the genetic variance compared to the full variance, because only genetics we breeder can influence. Therefore, we have to test it for each trait, because in each trait it's different. When we know the heritability, we can increase it by testing at more locations, with more replications in the field and with more years to be tested. Unfortunately, that costs money and needs a lot of seeds, which we often don't have in breeding. Therefore, we have to find compromises, and heritability is one of the major drivers for each trait to define the efficiency of selection. Most crops are grown in farmers' field. Therefore, we as breeders have to test our new products, the new varieties, in farmer fields. The climate and soil in different regions is different, and therefore we have to go in multi-location test trials, ideally across multi-years. In a single location, there are also problems occurring, because the trials are mostly large and there are differences in the soil, in the microclimate, or even in some herb pressure. To minimize these potential trial errors, we do a lot. We make special field designs, randomization of candidates across different locations. We score in the field when we see irregularities, and at the end we run statistical analysis trying to account for all that. It's evident, good field trials are the backbone of a successful breeder. The efficiency of our selection is largely determined by the genetic architecture of a trait. Therefore, we often run a so-called genome-wide association mapping study to understand the inheritance of the traits we work on. Roughly, we test 150 candidates in the field, and we make a DNA test with them with a lot of molecular markers. Running a bit of statistics, we can then elaborate the genetic architecture. When a single genomic region influences a large proportion of the phenotypic variance, we call it major QTL. And the technique we then use is marker-assisted selection. Unfortunately, most of our traits are inherited by many genes, and each gene has then only a small effect. These we call minor QTLs, and the selection strategy with markers is genomic selection. This is quite new in breeding, but gets more and more used. Together we breed for the QTLome with a lot of field trials.